Now we have 20 minutes for questions, so please answer your question, also mentioning who you want to ask. First question, please. Please.
Tore Scotland from Oslo. I would first like to thank very much Novartis and AstraZeneca for sharing their view on nanoparticle field with us. I think it's very important that people with that type of experience from pharmaceutical R&D give out their views. Secondly, I think that both what you told us and what we have learned during this Congress is that what this field really needs for the next years is more basic understanding of the biology, the interaction between nanoparticles and biological systems, what are nanoparticles doing to biology, and what is biology doing to the nanoparticles. So this is more a comment than a question, because I think it's really pity then when we learn what EU are going to use money for during the next years. It seems, at least to me, that it's more focused on late preclinical studies, clinical studies, and setting up pilot manufacturing facilities. But what we really need is more basic biological studies. So that is my comment. Thank you.
Any other question? Yes, please.
Yeah, this question might be relatively naive because I'm not an expert, but I can see that there are two types of problems. In cases where you know exactly where the problem is, where the tumor is, and in other cases where you don't know where the tumor is, let's say for metastases, if they are small enough. And don't you have to, and it's a question to all of you, don't you have to model your drug delivery and release system not just very specific to these two different types of problems? Because in one case you can inject locally and manipulate locally also, and in the other case you can't. And you somehow poison the whole body, more or less. That, in my opinion, is two different types of problems.
Yeah, I would tend to agree with that, and I think probably requires two different types of technologies. It would be great if we could take care of the tumor and metastases with one technology, but obviously I don't think that's available. And we certainly see some evidence out there now that there are technologies that go after metastases, and we're actively pursuing those.
Actually, indeed, such a product is available for treatment of micrometastasis. This was developed by Ciba-Geigy, the precursor of Novartis. And these are the MTPPE liposomes, now sold by Takeda under the name MIPECT. That's used for osteosarcoma, for treatment of kids having this disease. And this product activates macrophages, which are able to kill, to remove selectively metastasis in the lung of these kids. So such products exist and such strategies also exist, but unfortunately no one is really paying attention to this, I have to say.
Yeah, also in certain situations it's possible to inject into the arteria, that going to a certain area in the body, but it's limited nowadays to a few indications where it's really clear where to target, and obviously it's currently not yet the state where we really understand how to direct nanomedicines good enough.
I think, first of all, this table really have a lot of knowledge, and that's really impressive. But what I want to say is related to the lecture of Novartis work, that's actually typical to see what you saw, but we have a lot of other typical, much better things to show also. And I think that there are few major issues that we have to resolve, that without them we cannot really solve we cannot get this field to bloom, basically. I think Okay, now we know how to make the particles. We still face with some problems, but basically some people at least have the knowledge how to do it right in many cases. And we also know how to get particles to inflammation and how to get them to tumor. Not to all tumors, I agree, but to some tumors. Okay, and I think now with Jane and Maed a new idea about dealing with the matrix or dealing with the pressure and so on, we can improve on this. What we miss are really two critical aspects. One related to the targeting problem, and the targeting is a kind of sometimes you do, and I think Gert Storm probably and Terry Allen can comment on it more than I can, because I did a little bit of targeting and then I decided to leave it. And for reasons that there are a big unknown there. And the unknown is the following. If you succeed to get the particles to extravasate from the blood vessels, what they will do, they will find the first target and bind to it. And the first target are the first layer of tumor or whatever cells they see. Now, if they internalize very slowly, they will block the way for other things to come. So what you get, it's fantastic for imaging. We did wonderful imaging with very methods, and this was really a dream, but efficacy was sometimes poorer than with the liposome without the targeting, sometimes identical. Okay, so I think without knowing the kinetics of internalization, and I think internalization has to be very fast, not a matter, very fast, I mean a matter of minutes. Hour, 1 hour. If you don't, you may lose all the process. Then you may gain toxicity for many reasons. So, and the second thing, okay, for non-targeting liposome, so this is for targeted liposome, in which the mechanism is really uptake by the cells and through this activity. So you actually don't get good uptake by the cell because only the first cell get it, and therefore you may lose all of it. For the things like doxil or other free-floating liposomes, even if we ignore the matrix problem that Rakesh Jain pay attention to it, and I think it's a real problem, and now people starting to touch upon it, still you have to induce the release. Now you are in a very difficult situation. If you make liposomes that are leaky, they will leak in the plasma. You don't gain anything. Plasma, in a way, is a very aggressive environment. It has the lipoprotein, it has many other protein, it has some enzyme, it has, you name it, it has it. So, I mean, you lose the drug. If you make it too robust, then it will not release. So, what is the balance? The balance is to be based on the unique properties of the diseased tissue. For this, you have to do a lot of research now to understand what are the properties that may help you with your specific system to get release. I mean, I was lucky with the ammonium. Without this, doxorubicin will never be approved. We didn't know what's happening for many years. We knew that there is a release because doxorubicin was metabolized in the tumor. This we already found in 1992. How we know doxorubicin was metabolized is simply measure the metabolites, but doxorubicin never metabolized in plasma or in interstitial fluid. It must to have the internal environment of the cell to be metabolized. So it was obvious that one way or the other we get doxorubicin into the cells. And later we saw efficacy, okay, can be, you can argue how good and so on, but efficacy is definitely there, we know. So we were lucky. We didn't understand the mechanism. It took me, you can calculate how many years, from 1987 that I put the first patent on doxil till now. It's quite a long time. Till 1.5 years ago, we found the ammonium release mechanism. So I think, you know, and enzyme, I work a lot on enzyme, phospholipase A, and so it didn't do any release. Nothing, whatever, any pH, whatever we tried, and many other methods. So we can release with, you know, FDA, if you look at the requirement for generic doxil, they ask, do any method just prove us release and give you a list of about 20 things to do to get the release. Try in every pH, every temperature, nothing happens. Ultrasound release, we know it. I mean, we know ultrasound release, not a big deal. Actually, it's released by interest mechanism, not by breaking the liposome, but making pores in the membrane. The doxorubicin is released through the gradient loss, probably. So that's another interesting thing, but unrelated. But this, to apply it to metastatic tumor that's distributed is not do you any good. I mean, so I think these are the major issue that a lot of basic research, and I think company like Novartis should support this research with the young people and so on to get answer to these major questions. Without this, the area is dead, really,
sure,
unfortunately.
So I appreciate the explanation, and I completely agree with it. And that was my point, was just bringing awareness to this sort of community that in order to get traction, I think in big pharma, we're going to have to probably have a lot more understanding, do a lot more basic research. So I completely agree with that. And that's a two-way street. We need to contribute, and innovators like yourself need to contribute, and together we need to form something.
We just need the money.
Yeah. Yeah. And it's funny how that works because, sometimes the money comes with some little successes. And that's why we tried so many. I told you we tried maybe 12 different nanoparticles with almost all the same kind of results. Maybe that's a naive way to do it, but even if we had a glimmer of some success, and by the way, we still have some ongoing, so maybe we'll get there.
Okay.
Even with some traction, then I think the money comes. And it has to be just even a little bit
No,
of
I
traction,
agree.
the money comes.
We are not looking for free lunch.
Yeah.
We are looking for lunch that worthwhile.
Terry, please.
Hesse mentioned that I have a lot of experience with these ligand-targeted systems. And here's what I think. We've done a lot. I mean, most people work on these primary solid tumors. It's not what kills the patients. What kills the patients are the metastases. And we've done a lot of metastatic models and pseudometastatic models, and what we find is that these ligand-targeted systems are really quite excellent in these metastatic models. They're very good at getting to the very small metastases. Where the problem comes is the fact that it's very difficult to do clinical trials in a metastatic setting. You have to do an adjuvant trial. And the system is a little biased against doing initial adjuvant trials. And so you have to normally start out with patients that have a very high tumor burden and whatnot. The ligand-targeted liposomes with a variety of drugs and targets do not do well in a primary tumor setting. They do really well against these micrometastases. And so until we can actually start testing them against this metastatic disease, I don't think we're really going to be in the right niche for them. But in the meantime, we're doing all of these experiments in subcutaneous models or xenograft models or orthotopic models, but always against these big primary tumors. And, the clinicians surgically handle it mostly by going in and removing the tumor. Those are not the things we need to be treating.
Yeah. So for me, I mean, reviewing the last few years of Clinam and going through today's sessions, I think I see also in review papers showing up that more and more rules for success or trials to avoid are published, and I think this shows the momentum that more is understood, which will probably help to focus the efforts to the situations of easiest translation, if it's easy at all. And it's easy to get it wrong, absolutely. So, but on the other side, I mean, we saw this nice section from Hesse about the characterization, also the NCL efforts to have assay cascades, start with some critical tests, and then expand it once you approach to maybe first human dose. And then Terry's summary on what situation would be easiest to investigate with nanoparticles. I think this frames the situations. Now, the next question I would have is, we heard from Heiko what the EC is going to fund, and I'm wondering whether you would explain a little what kind of situations for, let's say, engagement for industry or collaboration in consortia in general for the community you would propose work best.
Okay, so for the pharmaceutical industry, is certainly IMI2 the best forum to discuss about this? Because IMI2 is a joint undertaking, joint technology initiative, public-private partnership between the EU and the pharmaceutical industry. So especially FPA, who both contribute about 1.6 billion euro. And so the priorities are set by the members and the board about what's going to happen. And so far in IMI1, targeting was never a priority. I think there have been some calls, but it was never a priority. So the chance is to say, well, we want to do more on nano medicine in IMI2. And to apply nanotechnology there in the projects. That gives the possibility to try out with a forum, the cost and the risk are shared.
And what about the other, let's say, NMPs or so that are already underway or just starting in the proposal phase? I mean, is there flexibility to get in or out? Or
also there. So in the moment where we have the working program 2014 and 2015, but the priorities for 2016 and 2017 will be coming up next. So we will be discussing about them, because each time the priorities are set for 2 years. So the next period will be 2016 and 2017. And then it's important to think about, as a nanomedicine community, as a pharmaceutical community, what do we need? What is our experience from all the work that has been done so far? And how do we want to continue in the future? What are the priorities? So for the CATS, where I am, the Industrial Technologies Program, we've said, well, we've done a lot of basic research in laboratories, with mice, and there you see a lot of good results, really. The projects that are running, they work according to their objectives and they get results, and sometimes excellent results. But then the problem next is to make bigger quantities of nanopharmaceuticals and other, say, nano components. I heard even in the micro-nano biosystems sphere, the difficulty is with the manufacturing of pilot quantities. So people make one system in a laboratory, but then the next time they need to test more systems, say 30, 40, 50, and there is no capacity to do that. So I think that all these things need to happen in parallel. We need to increase our knowledge about manufacturing, characterization, and at the same time there were other comments that the biology should be improved. Well, that should also be done, probably. That should also be done. So but in the end, once you understand the biology, you say, well, now I want to apply, say, a therapy to this link somewhere, and then you don't know how to manufacture your pharmaceutical. So you have to work in parallel. So that by the time you understand what you need to do, you have a manufacturing process that you can use to apply it.
Jose, please.
Thank you very much. I unfortunately was coming here in the middle of your talk. You mentioned the IMI2. I'm not aware of any specific nanoparticles or nanomedicine group, but what I can tell is that there is a scientific governance group for immunology that is now being formed. And these have many diseases that they are basically the target of the nanomedicines that we have been discussing these 3 days here. And there is an opportunity to bring basically this as a kind of important component of this group. And maybe this can help, and it can help everybody.
Right, right. Okay. Yeah, so, and then the other forum where you can discuss this is of course in the health program, which is the medical program, and which has by far the biggest budget for medical research. And they have quite open calls. For instance, a call for diagnostics, you know, that is quite open for all the possibilities, and there the nanomedicine community can then submit proposals. And well, if there are very good proposals and the technology is powerful, then we may expect that those projects will be selected rather than, say, projects that are less advanced with our ideas.
Okay, any other questions from the podium? Yeah, okay, so I think then we can close the session. Thank you very much for the vivid discussion. I think this conference has been very good and even better than the last years, and I would also like to thank Patrick and Beat for making this very unique conference so special and attractive. Thank you. Bye-bye.